Welcome in alessandro-project.
Ligand-based alignment Cidofovir library natural compounds. Results have greater analogy for the reference 2-deoxycytidine 5-monophosphate used for the treatment of leukemia. Cytidilic acid, 5-methyl-2-deoxycytidine. Sulforaphane present in many species of the family Brassicaceae. Are present on PubChem. Many studies on the clinical use of Questa molecule and its antiviral activity has been demonstrated on several variants of coronavirus, 5-methylcytidine and cytidine. Alignment Cidofovir library antiviral compounds results. Lamivudine and ensitarabine obtain the greatest analogy to the reference. Structure-based 1. DNA docking, PDBID2L8P. Own antiviral compounds results nephrin nootropic with antiviral effects SARS-CoV, antineoplastic nilotinib, kin 1148, modulating the antiviral immune response. Camptothecin is an antineoplastic topoisomerase inhibitor. Dinosaur inhibits clathrin and dynamine-dependent endocytosis in cells. Cell sept. Gandotinib Harno obtained the best scores. Docking on DNA, PDBID2L8P. On natural compounds library results best scores for nephrin. Halofungin OWN is a coccidiostat used in veterinary medicine. Sedative anti-inflammatory magnoflurine and natural anxiolytic. The antiviral activity of nephrina is confirmed by Yang et al. 9. Docking on small polysomerase topoisomerase. PDBID3-exatii of the compounds contained in cocoa. Theobroma cacaoel. 1753. Table 1. Cocoa compounds have a high affinity for the two topoisomerases considered the structures of catechin derivatives. Screenshot 5. Catechin, epicatechin, gallocatechin, GC, epigallocatechin, EGC, catechin gallet, epicatechin gallet, gallocatechin gallet, epigallocatechin gallet, egg-gay. T with scores similar to co-crystallized, as confirmed by Yoshida et al. Docking of St. John's wort compounds. Hypericum perforatum on small polysomerase topoisomerase. PDBID3-IGC. Hypericum compounds are active on topoisomerases, as confirmed by Catherine et al. 6. The Atibius principles of St. John's wort. Procyanidin B2, pseudohypericin, mentoflavone, hypericin, isoquercitrin, and biapigenin have a high affinity for smallpox topoisomerase, demonstrated by the scores. Screenshot 6. See screenshot caption. Procyanidin B2 pseudohypericinia mentoflavone hypericin hyperoxydisoquercitrin biapigenin. Protein alignment the proteins topoisomerase of S. Pneumony, PDBID3 reeti, and topoisomerase of the smallpox virus, PDBID3 exatii. Figure 4 additional are aligned. This is necessary, as in the crystallized smallpox protein, there is no reference drug or molecule that highlights the possible binding pocket, while levofloxacin is linked to the topoisomerase of S. Pneumonia. Alignment of the two chains. They are equal to 15.56%. Figure 3 additional. Covalent docking on L1 Poxvirus protein of natural compounds containing nitrile groups. Table 2. And drugs containing nitrile compounds results many store in obtain the score some more alter. Considering the electrostatic complementary E0. DNA docking. PDBID2L8P. Blaze server Chembl 1,213,016 is the compound with the greatest analogy, but it does not have a toxicological profile, nor a studied biological activity, or a clinical use. And the same for the first 7 classified.
Conclusions. In 2022, there have been cases of smallpox infections, despite the fact that smallpox has been completely eradicated since the 80s and there are no drugs currently available, excluding Cidofovir, which has marked side effects that are able to counteract the systemic infection of this virus. This study aims to search for natural compounds or drugs with known toxicological profiles that could counteract smallpox infection. Drugs currently in use that could be effective for this pathology and have obtained good scores include 2-deoxycytidine 5-monophosphate, a reference drug used for the treatment of leukemia. Antiviral drugs such as lamivudine and ancitrobine, nephrin, a nootropic with SARS-CoV-2 antiviral effects, antineoplastic drug nilotinib, KIN 1148, which modulates the antiviral immune response, camptothecin, an antineoplastic topoisomerase inhibitor, and Dinosaur, which inhibits clathrin and dynamin-dependent endocytosis. Vilazodone, used to treat major depressive disorder, is the drug with the highest scores, binding covalently to serine at the entrance to the pocket and occupying it, see additional figure 8. Other drugs include neratinib, a tyrosine kinase inhibitor anti-cancer drug, BMS 191095, which opens mitochondrial potassium K, ATP, channels, is cardioselective, and inhibits platelet aggregation, and epanolol, a beta blocker.
Natural compounds, omitted from previous studies, have similar scores to the reference drugs. Sulforaphane, present in many species of the Brassicaceae family, and in food supplements. Allein, artemisic acid, present in the genus Artemisia. Halofuginone, a coccidiostat used in veterinary medicine. Sedative anti-inflammatory magnoflurine, and natural anxiolytic. The proteins of catechin derivatives are found to have a high affinity for smallpox isomerases. See screenshot 5. These include catechin, epicatechin, gallocatechin, GC, epigallocatechin, EGC, catechin gallet, epicatechin gallet, gallocatechin gallet, and epigallocatechin gallet. Egg-gay, menistorin, a natural nitrile compound, would bind to tyrosine covalently. Other compounds such as falcarinone, falcarinol, xanthohumol, rosmarinic acid, vitamin C, vitamin K3, and adhumulone would form covalent bonds with nucleophilic residues, according to their scores.